Muse cells versus stem cells. So in this video, we're going to talk about what it means when someone says stem cell therapy. They could mean embryonic stem cells, it could mean induced stem cells, it could mean mesenchymal, stromal cells, or Muse cells. And so we're going to talk about the differences in safety, mechanism, the clinical applicability between the two, because these categories, there's an enormous difference between these two types of cells. What are Muse cells, or multi-lineage, undifferentiated, stress-enduring cells, versus mesenchymal stem cells that were renamed by Arnold Kaplan, more appropriately calling them medicinal signaling cells. And so the context is really important. And then when you start inducing some of these stem cells, especially if you're using like these pluripotent or totipotent embryonic stem cells, this is where there can be massive differences in what they do, what it means, and the safety of your health, which is the most important thing. And that's why this channel is made. I'm Regan Archibald. I record videos like this because I see a lot of unhealthy people and people who want answers, but they don't know what direction to go. And so I'm the founder of Ageless Future. We've been in practice for 22 years, and we've helped over 70,000 clients, people who are high achievers, high performers, looking for that next level in their health, but they don't want to risk their future health. They don't want to risk results by being on certain medications that are going to just suppress symptoms. They want an operating system. And so for those of you looking for an operating system, go to agelessfuture.com. My team, we're all trained in functional medicine, and we'll run the right labs and figure out what is interfering with your future and remove those interferences, and then help you build the best health of your life. Let's talk about Muse cells and MSCs. So here's the big issue. Anytime you start replicating cells, what's the risk? Well, if you have like these embryonic stem cells or these pluripotent stem cells, then these induced pluripotent stem cells, they can differentiate into virtually any cell type in your body, which you'll say, well, that's what we want, Regan. Well, the problem is when you're using these from an external source, they can form teratomas, like these are growths. They can form tumors, and they can form these disorganized tissues from multiple germ layers. And in essentially 100% of the immunodeficient mouse experiments, it happens. And so this is not some fringe finding. This is not like the FDA trying to like suppress the outcomes of stem cells. This is the results of 25 years of research. And it's the primary safety barrier between, from clinical translation where, these cells are used, them being just kind of in the background of just research use only. But here's the interesting thing. When you look at these stem cells and you look at the reprogramming, first of all, like what are stem cells and why are they there in our body? Well, stem cells, specifically your mesenchymal stem cells, these are the workhorse reparative cells. They're found in your bone marrow. You'll find them in your adipose tissue. And they do have a very large safety profile as long as these cells have not been more than minimally manipulated. That means, you take out a fat transfer and you spin out the stem cells and then you can re-inject it. But the problem is when you induce these cells and you start feeding these stem cells and culturing them so that you grow more stem cells, that can be dangerous. MSCs, they can also come from the umbilical cord or the placenta tissue. And these signaling cells, they do seem to have very robust regenerative properties. But once again, if you take these cells and you start expanding these cell lineages, that's when the risk starts to increase and starts to multiply. And this is where there's been such a hard line between what the FDA classifies as minimal manipulation versus not. Now, in Utah, we have a bill that was passed in May of 2024, Senate Bill 199, that protects us in using these perinatal stem cell tissues because the medical board in Utah says as long as you fits the FDA criteria of minimal manipulation, there's not marketing claims being made. So once again, they are not FDA approved. Just because they can be used clinically does not mean they're FDA approved. But as long as they meet the criteria, and we're working with these registered labs, the labs we work with and that we've worked with over 15 years, these are healthy mothers undergoing a cesarean birth. This is their first baby in many cases. And we do rule out any mothers who've had COVID vaccine that can actually get into the umbilical cord, and we don't know if that could be damaging. So that's part of the screening process, looking at the mother's health, the father's health. And then once that tissue is donated, donated, what we found is to maintain a high viability of the MSCs, you need to have about a 20-minute window before that kind of rubbery tissue gets turned into a flowable. And then once it's turned into a flowable, 20% of that tissue gets sent to 2 third-party labs to be tested for any communicable diseases, anything that could pose a risk, and then cellular viability. And then once it passes that quarantine state, then it can be released. And so if we think about how does MSCs relate to Muse cells? Well, Muse cells are interesting because they too are in the umbilical cord tissue. They're in the placental tissue. They're actually in your body as well. In the human body, about 1 to 3% of all the stem cells circulating in your body are what are called Muse cells. Now, Muse cells just happened to be an observation by Dr. Dezawa's team in Japan. Mari Dezawa, she was looking at these fibroblasts in skin. She also noticed that these Muse cells were stress enduring. She found that when there is a harsh hypoxic environment, she found the Muse cells actually survived that environment. And then she started looking at all the tissue structures, and sure enough, she found that Muse cells were there. And so what we found is even in the umbilical cord tissue, some of the most robust differentiation, some of the most clear regeneration properties probably come from these Muse cells that exist in the perinatal tissue. And so one thing we're looking at is, well, what's the difference between a Muse cell and an MSC, and which one's better? And so I can tell you from a Muse cell perspective, Muse cells do have pluripotency. So they were able to isolate the particular markers that would show that a cell is pluripotent. And Muse cells differentiate into all 3 germ layers, which is very unique. They also found that Muse cells, they don't form tumors or teratomas, and this research has been done over the last 16 years. In one of the studies called Cellular and Molecular Life Science 2023, they found what appeared to be paradoxical, because it's like, how can a cell like a Muse cell express pluripotency genes without forming a tumor? Because that had never been seen before. And what they found is that this protein that is suppressed, and in the ESCs and the induced pluripotent stem cells, or ESC is the embryonic stem cells, they found that the RNA binding protein called Lin28 suppresses tumor suppressor miRNA LET7. So without LET7, these pathways run unchecked, allowing unrestricted proliferation, which means teratomas and cancer, where you can get tumors. Muse cells do not express the Lin28. They express LET7 at higher levels than induced pluripotent stem cells. And LET7, that allows these Muse cells to maintain pluripotency without turning on the genes that would trigger like teratomas or tumors. So very interesting. And in Japan, they're looking at it for ALS. They're looking at it for Alzheimer's. They've found with dementia studies, even studies on myocardial infarction. So if you think of Muse cells, one thing that was unknown is how do they differentiate? So what is the mechanism of action? What's the signal that's turning on the Muse cells to become pluripotent? Because you put one Muse cell into a tissue or into a body, and then that cell doubles and then triples. It's doubling in number every day, and it's like this exponential curve. But the way they discovered is through a process called phagocytosis, where the Muse cell comes in and takes in this underperforming cell in your body. They classify it as an apoptotic cell, that your body is, pre-programmed cell death. And it takes the raw material and remodels the cell, and then it goes on to differentiate and create the regenerative properties that your body needs without running the risk of that teratoma or tumor. And so I'm very excited because I see in medicine today, we're learning more and more about what our body does to regenerate. What do we need to do? And the exciting thing about Muse cells is the studies that have happened in Japan. These have been going on for over a decade. And so I'm excited to see us bring these into the United States so that we can see how we can help those of us who want to live as long as possible in as young of a body as possible. I think Muse cells will be at the forefront of it. So at Ageless Future, we work within this MSC and exosome space. I think the Muse science is something that I'm very excited about. And so for those of you who want to go deeper, find out if this is right for you, go to agelessfuture.com. My team will take great care of you.